I'd like to welcome you to our webinar series, the Human Islet Research Network webinar series. We have two fantastic speakers today talking about an exciting topic. We're going to get into that in a minute. Some of you have become accustomed to joining these webinars. Maybe this is a first for a few of you. So I just wanted to introduce HIRN to those of you that might not know about it. HIRN is a collection of different groups that are working together with the goal of trying to understand the pathogenesis of type 1 diabetes and trying to come up with different translational therapies that will eventually be used to help people that actually have the disease. And the way this is done is it's been organized around central themes. And what you see here to the left was how HIRN started in the beginning. And these were several different themes: consortium on modeling autoimmune disease, for example, and the consortium of human islet biomimetics, and the consortium of targeting and regeneration, and then the human pancreas analysis consortium, and the consortium of beta cell death and survival. And then there is kind of the organizational hub, which I and others are a part of, the Human Islet Research Enhancement Center. But HIRN wasn't ever meant to be a stagnant network, and it's evolved over the years, most recently to what you see on the right hand of the chart. And if you're interested in any of these, you can look up the publications and get those. But essentially over the years, the 11 or so years that we've been in operation, it's been very productive, not only in terms of the number of discoveries that has happened, but just in terms of the number of researchers and trainees and number of institutions and people globally that have been involved in this initiative. And if you want to know more about this, you can also sign up. We have a newsletter. You can subscribe to that newsletter, and you can find out about funding opportunities, about different discoveries, as I said, that have come up, different opportunities, jobs, people post openings for postdocs and other things there. So a number of opportunities. And you can also do that by going to our website as well, which I'll tell you about. Now, in terms of the funding opportunities, briefly, I'll mention that there are two. There is a Data Scholars Award initiative. HIRN has generated a lot of data and wants to encourage the community to use that data in novel ways. We just recently had a submission deadline pass, but there's another one coming up in November. So you can look on our website and find out the details about that. There are a couple of different initiatives under this Data Scholars program. That's one. And that is a funding opportunity directly coming from HIRN. The other one is from NIH, and these are called Gateway Award initiatives. You can find out more about there, but essentially this allows you to come into HIRN through NIH funding, and you can become a part of any of the consortiums that I just briefly described to you. This is our website. Again, all of what I'm talking about is there listed on the website, particularly the funding, which you might be curious about. You can find out more there. But we have a lot of other resources and things that you're not going to hear about today as well. And so you can visit the website to learn more about those resources and also learn more about resources that we have with our collaborators as well. You can touch us on different social media websites as well. And again, all of the resources, so programming resources, antibodies, constructs, different types of animal models that are being used in HIRN are all cataloged here for you to view as well. And again, you can get to that to the website. Don't worry too much about writing down anything as you listen to the presentation today. All of it is recorded. We post that on our HIRN YouTube page, so you'll be able to go back and revisit that should something pique your interest. And so with that, I'm pleased to put this presentation to our speakers. Just a few reminders. Again, make sure that you're on mute. We will save questions until the very end, and we anticipate that we'll have a good number of questions that we can take at that point. And so with that, I'm gonna unshare my screen, and while I am unsharing my screen, I'm gonna invite Wenbo and Zuwan to put up their screen as I introduce them. Zuwan is a postdoc fellow, and Wenbo is a PhD candidate, both at Harvard. They're both part of Jiao Lu's lab at Harvard. He's an assistant professor there. And they, as being engineers, have been collaborating with Juan Alvarez's lab at UPenn. And they've been both working on this very interesting project, which we're going to hear about today. So, Zuwan, please take it away, and I look forward to your presentation. Take yourself off mute, Zuwan.
Okay. Yeah. Can you everybody see the screen? Can you see the screen?
Yes, we can see your screen and we can hear you
Great.
well. Thank you very much.
Great. Thank you so much, John, for the invitation, and thanks for Hern for the invitation as well. So, hello everyone. My name is Zuwan Lin, and this is Wenbo Wang. So, we work both in Harvard Seas and the Broad Institute with Professor Jia Liu to develop the agentic AI system for studying a wide topic of biologies. And we also collaborate closely with Professor Alvarez and Professor Wang and Professor Lina to apply this agentic system to study the biology. So today we are very excited to share our recent work about using the AI agent technology to adapt it, to apply it in the spatial transcriptomics field, and then use it to further study the human islet systems. So now let's get started. So in today's presentations, I will first introduce the concept of AI agent and how it works, and then Wenbo will then further demonstrate the more technical details about how we build this spatial transcriptomics AI agent and apply it to the pancreatic system. So now let's get started. Um, So before we jump into the main topic, let me first introduce the concept of the AI agent. So AI agent has been a topic for more than decades, and in machine learning and artificial intelligence, it has usually been defined as intelligent entities that can perceive its environment, and then based on the perception and observation, and then take actions on the environment. During this process, the state of the agent and the state of the environment will get updated. So this may sound a little bit abstract, but if we view ourselves, we human are also like an agent because we can perceive our environment and then get feedback from the environment. And based on this feedback, we take action on the environment. And if we put the agent in the context of a physical environment, we will have the examples of the robots and autonomous car. And if we put the context of the agent in those digital gaming environment, we have examples of AlphaGo and AlphaStar. So these are very remarkable agents, but they are really highly skilled and task specific and non-generalizable. For example, you cannot expect AlphaGo to help you analyze the biological questions or like the AlphaStar to help you think, brainstorm the scientific ideas. And with the recent rapid development of the large language models, it has really added remarkable additional action layers to this previous mentioned agentic framework. These additional powerful layers is the reasoning and planning capability of the language models. It can be viewed as internal actions of these agentic systems, just like we human will do. We human, when we think, we have a kind of inner monologue. So this LM actually serve as the brain of this new AI agent system. So what is the main advantage of this agent system compared to the previous agent paradigm is means that it is really generalizable because you use the LLM as a brain. So actually, its action space can be viewed as an infinite language space. And since they also natively understand the human languages, the human can also directly communicate with the agentic systems through the natural languages. So these adaptation of the language agent has really bring the traditional agent field to a new frontiers. So let me briefly introduce what could be the key capabilities of LM in the AI agent development. So the LM has really showed a remarkable capability in terms of reasoning and tool use. In the reasoning, it's actually quite like what we human will do. Like it can do a lot of planning, decision makings, and calling other AI agent, calling other AI tools. This part is really like what we human will do. Like we cannot solve all our problem by ourselves, but we know how to use tools to solve the problems. For example, we cannot perform really complex arithmetic calculation directly in our brain, but we know we should use the computers or calculator to do that. This part is really like the reasoning capability of the agent. So it can actually decide what tool to use at what context. And in addition to this reasoning capability, it has also showed a remarkable capability in codings, in context learning, and also parsing. And these tools is actually really important for the LLM to become an agentic system because through these tools, these LLM can actually start to interface with the digital world. And given this context of the LLM and the general agentic systems, the major challenges for really building the scientific agent is actually the building this what we call the domain-specific action space or the tool set, and also the domain-specific perception action logic. And this is through scaffolding and prompting. So this tool set can be viewed as like how we human will perform things. We'll first like start to build a tool first and then utilize these tools to solve the problems. And for this domain-specific perception action logic is really like how we teach a new grad student or new trainee about how to do things. Like we will give him some instructions and examples about given some scenarios like for these scenarios you should do this and then use that tools to do that. And the LLM itself doesn't have knowledge about all the domain specific problems. So that's why we need to build this agentic AI system to really make it useful to solve the domain specific problems. So our research has long been concentrated in developing this agentic system for different layers of the biological data analysis. So these different data layers can span from the molecular layers, functional layers, and system levels. And all these data can be viewed as the environment that the agent actually perceives. And since that this data is with various data format, like the spatial omics data, it comes with transcript expressions together with its spatial location. And this functional data is with time series data. And behavioral data could be those video data. And all these data really requires different action space to for the agent to interact with this. So a large portion of our agentic AI work is actually trying to build and connect all these action tools to the agent, and then teach the agent how to use these tools to solve these specific data problems. So in today's talk, we will be mainly focusing on how we actually build the spatial transcriptomics AI agents. So here shows actually a very high level interview of these agentic AI systems. We actually have the agent using the LLM as its brains, and then the spatial omics data is the environment that the agent interact with, and the here also show some representative tools, which is the agent's action space. So these are some of the common tools that we will use in our routine analysis, and the agent don't have an idea, okay, how to actually use these tools, and we need to make the agent be aware of the how to use these tools. For example, the literature search tool, the packages for analyzing spatial omics, the single cell omics, and also the codings, and as well as the report generations. And we also need to manage the memories of the agent for agent to not lose the context of what's the data itself interface. And next, Wenbo will talk more about in detail how we actually utilize this system to study the pancreatic biologies. So.
Okay, thanks, Zuwan, for the comprehensive introduction on the AI agent system. So now let's come back to the details about how we apply this AI agent to the spatial transcriptomics things. But let's go through the spatial transcriptomics in a brief introduction. So within the past around the decade, people start to realize that for single cell biology, it's not enough to only study the individual cells' molecular features. On the other hand, how where each cells are located, how cells are interact within the tissue organization context is also very important. So for the past 5 years or 10 years, people starting developing this kind of in situ sequencing technology, which they can preserve both the locations of individual cells together with capture its molecular features. And this will yield this kind of data structure with the gene by cell matrix together with its XYZ coordinates. And if you see that analyzing this kind of data set is very challenging. So first, it needs so much of the domain-specific expertise on specific data questions, which needs around years of training. And also, the data set itself very complicated to analyze. They have so many covariants, they have so large matrix, and you need to plan detail on each task, what aspect of the data set you are going to look at. And this one, the intrinsic multimodal means that you don't only have this numerical data and the description, you also have the images, which you show how the spatially organized our individual cells. And it's also contextual, means that for different questions, different organ, different projects, need to have context-specific information on it to generate the specific pipelines. And actually, this kind of things are well suitable for using the AI agent to do analysis, as I mentioned, for the following reason. First, the agent have this reasoning capability, which it can based on the current context about the instruction, the prompt to make a decision on what the specific task they're going to perform and how you're going to tackle the questions. And the LM also have this multimodal handling capabilities, which allow it to not only take into consideration of the text, also be able to analyze the images by through this image encoder and embed into some image tokens. And output will be in a text format which can be easily understood by humans, which means we can regard it as explainable analysis. And the last, which is the most important, is this extensible action space. As I already mentioned, that the one very charming feature for agent is to leverage these different kind of tools. For example, the web browsing tool like Google Scholar or other web browsing, or this kind of well packages like ScriptPy or ScanPy for generating specific pipelines of analysis. And they also have this coding capability to execute the code so that they can build a data set specific pipeline for analytics. And they also have the memory and report system. So that is our inspiration. And then we decide to develop this STAgent system to have this different component for make it a potent AI agent for spatial transcriptome analysis. It has this multimodal contextual memory to store the analysis results. It has this AI agent orchestration framework, and it also has equipped with this complicated real-time literary research tools and also the code database that can retrieve from them to generate data-specific pipelines. We then try to apply this system by collaborating with Professor Alvarez on this pancreatic system. So let's see our data set to apply for. We studied how this stem cell-derived pancreatic graft mature in vivo. So we transplanted this pancreatic graft onto the mouse kidney capsule, and we harvested them at the relatively early stage, like 4 weeks. and the relatively later stage, like 16 and 20 weeks, then perform the in situ sequencing. We leverage the technology we previously our lab and collaborator developed called StarMap, which is a 3D spatial transcriptomics that can convert this individual mRNA within cell into DNA amplicon, and we can perform the segmentation to reconstruct the whole tissue architecture. Uh, just briefly introduce technology. To apply the technology to the pancreatic system is actually very challenging because the pancreas are intrinsically abundant with this ribonuclease and proteins, which will stop you from building a very comprehensive DNA library for these different genes. So we optimize the StarMap protocol to including this denature part to effectively remove the endogenous RNAs and DNA and the proteins. And then we successfully have this kind of like sequence result on the pancreatic tissue. You will see each individual color shows the individual amplicon that represent the different genes, and we can align them through multi-round of sequencing. And the result you can see from here that we capture the different stages of the pancreas graft maturation on the mouse kidney. So that's the data set, but the important thing is that how we can apply our STAgent to analyze this data set, right? So we're starting by testing its autonomous code generation for the analytic pipeline processing. You will see from here that we can just use the natural language query to instruct the agent to generate this analytic pipeline by performing the code generation execution, generating the meaningful plotting result, and then try to analyzed by agent themselves. So as an example, you will see, I will just say that please help me perform end-to-end analysis on my spatial transcriptome AI agent data set, and then I can generate a report. So the agent will start by looking at how we can plot in this UMAP embedding, look at what's the difference between this week 4, week 20, and week 16. And then it will continue to do this temporal cell composition analysis changes, and also the cell and also the spatial distribution plot, followed by some like a spatial neighborhood enrichment analysis. The thing we mentioned that this all it are able to generate this different kind of the results, but also it has this VLM capability, which means that it can take this plot figure into themselves to perform a exhaustive investigation. So here shows the example about the visual reasoning capability of the agent. So the agent autonomously take into this plotting of the early stage spatial plot of different cell types and also the late stage spatial plot of different cell types. And they can reason across images to get some biological plausible implication. For example, it will look at that, so at the relatively early stage, the organization of the alpha and beta cell seems like a scattered and a non-organized way. However, at the later stage, you will see they form a kind of like co-shell structure, which more recapitulated the mature islet structure in biological models. Another thing, as an example, is showing the expansion of the mesenchymal cell population, which you can also see the expansion of this blue cell, which is the mesenchymal cells, and also the green cell, the mesenchymal cell. They significantly expand from the early stage to late stage. The agent can autonomously capture these spatial features from different stages and generate some biologically plausible implications. And more importantly, the reasoning results can also be paired with this real-time online literature knowledge search. So the agent will autonomously perform this action on each individual biological implication to search for the literary references that can prove or prove against it. We get the, for example, if we from the different time point of the spatial plotting, we can generate this different biological implication like the add architecture evolutions, the revascularization patterns, and also the expansion of the mesenchymal and the asynchrone cell populations, also the host graft interactions. Another feature we are trying to decide about is the context-aware gene set analysis. So that's related to the third challenge we mentioned about the spatial transcriptome data set. It's about the contextual nature. For different organ, different projects, you need to consider different spatial context. Right? You are under the pancreatic contact, you are not just blindly put all the genes there and see what's the potential interactions. So traditionally, people will just like, for example, run the whole gene groups that are elevated and then generate this enrichment pathway and find and the human to curate that. If this one is related to the pancreas biological process or not. But now the agent is able to autonomously take into consideration this different gene set and find which part is related to related to the pancreatic context, and then they will perform this literary research to search potential implication, potential biological discoveries, and then summarize into the context-aware gene analysis. This example, we just ask the STAgent, are able to look at how the beta cells are different from the early stage, like 4 weeks, to the later stage, like 20 weeks. The agent will automatically generate this code for finding out the elevated, upregulated genes, and then try to look at the individual gene set to find some if they are related to pancreatic context, and then generate this biological implications. And finally, the agent will try to formulate into some bullet point that will tell you about what happens during this pancreatic maturation process in vivo. Finally, I want another thing we want to discuss about is the deep research generating capability. So we mentioned this different kind of section that different part agent can perform. For example, you might be embedding generation, cell composition changes, and content-aware gene analysis. However, each individual point is kind of isolated, right? So, for example, when you consider you are writing a paper, you're doing different kind of analysis, but when you write a paper, you need to blend them together to generate a coherent story or coherent deep research like paper to tell you the whole system. So the agent is able to leverage this different part of analysis pipeline and then iteratively search for the knowledge base, for example, Google Scholar and other local databases to generate the coherent deep research-like reports, which include the objective, overall of the study, key findings, and conclusions, just like what you read in your paper. So this is an example showing that it's taking into consideration of all the previous analysis on its, for example, embedding analysis, population dynamics, and also the gene set analysis. And then they blend them together, generated bullet point of the biological implications, then go through one by one through its iterative retrieval to finally generate deep research-like reports. I can briefly show you guys an example of how we run this end-to-end analysis. So this is an example we recorded a few months ago. So
Hi,
you
can
can
you
just
tell me
include a query like
end analysis on my spatial transcriptomics dataset, and then please generate a report. Thank you.
So you will see that the agent will take into this input and it will start to perform this analysis by formalizing the questions and decomposing into some different tasks and run the pipeline one by one. You will see that it plots this UMAP, and each individual plot will be taken to the agent and they will generate these implications and then start to use the Google Scholar to retrieve related information from the literature to see if you can find some evidence to prove that or against it. And then it generates the conclusion for the biological implication based on these researchers. Yeah, and this is the example of showing how the code are generated. This all code are generated by the agent and they will execute it with the Python repo tool. I'll just and then come to spatial plot of the data. I will just skip a little bit. And finally, after all this our analysis pipeline are finished, which is designed by the agent, it will just blend all this in together to infer from each sections to generate this coherent deep research lab report to supported by the literature searching and enhancement. Yeah, you will see here. And finally, it will generate this deep research lab report that tell you for the whole story from beginning, like what it have already performed, what the funding is found, and how to, where if there are any literature research that can support or for or against it. Yeah, with the references. So. Yeah, so here is the take-home message. So STAgent, I think we think the most exciting part is STAgent can shrink the spatial transcriptomic workflow from weeks of effort into some minutes of autonomous analysis, which even don't require you to have too much of domain-specific expert technologies. And this is very important for applying for example, a lab we are trying to study a system they are not familiar with. They don't need to like train a trainee from scratch for many years and start the project. They can just use this workflow to perform autonomous analysis generated by agent. And also it can generate some like for the meaningful biological implication from this pancreatic graph dataset. For example, the alpha beta structure involvement, stronger endocrine interactions, and methylome expansions. And also the third one is that STAgent can connect different isolate analysis pipelines into some coherent deep research reports that can directly transfer from the raw data into some structural output reports. And if you are interested in the paper, this is the link to the paper we published that on bioRxiv and now it's in revision. So I finally want to give the acknowledgement to both Professor Jialiu, who really lead the project and give the whole supervision, and also Professor Alvarez, who contribute a lot of the pancreas part discussion and also providing the samples. And also for all the Liu lab members and our lab members and also the Production lab members who contribute meaningfully to the project. So yeah, that's it. Thanks for listening and happy to answer any questions.
Yeah. Hope we didn't exceed the time. I think maybe the most important part for presentation is for the discussion part. So
Yes, that is. Thank you. Thank you very much, guys, for giving us this wonderful overview. What is a lot of work, undoubtedly. So if you have questions, you can most certainly raise your hand. If you would like to ask these questions yourself, we welcome you to do that. I do see a few hands raised, which is great because I know this is a really interesting topic and hot at that. So why don't we first take a question from Yao Quinn? I hope I said that right.
Yeah, Yixun. Yeah, thanks. Yeah. Okay, so I'll just quick. So one quick question. I see the app is running locally. So my question is about will this app be available online, or there is a GitHub repo so that everybody can clone and run their this app on their local computer?
Oh yeah, we have actually open source the current version of the code in GitHub, so everyone can download and start playing with it. And we also have include our demos data set, so you can go to the paper and directly check a GitHub repo. And one thing is that the current version we are still experiencing a major upgrades. So, but you can definitely start with like our original version. I think start to explore like how it can do.
Okay, so would you mind sharing the available link in the
Of
chat?
course.
Yeah, thanks. Really appreciate it.
Okay.
Let me share.
Terrific.
Let me share.
Thank you. Thank you very much for that question.
Maybe we can, should we stop sharing and then we can find
And you're welcome to send me the link. This is Layla, and I will send it to all the registrants after the meeting as well.
Yeah, that would be even better, probably. Yeah, we will send you the link. So maybe after the meetings, or we can share in the chat as well.
Okay.
Yeah, you may need a OpenAI API key or Ansible API key to do this. That's
Okay.
Yeah.
1 second.
Terrific. Thank you. Mina?
Yeah, right.
Hey, that was remarkable. I could imagine this shortening analysis timelines and super cool. So one question is a lot of the times it seems like you have to direct the questions to the agent, which makes sense. It's software, right? But is the agent capable of like, because you're asking it something, you're sort of directing it. How capable is the agent of telling you, that you might be missing something? Like, let's say there's some weird macrophage subset that you don't know that it's necessary for the engraftment procedure that you never would have guessed. So
yeah, that actually really, I think the agent, it really has the capability to do that. For example, we are probably not we are not trained in the pancreatic biology field, so we are actually in a perfect position to answer that questions because everything agent find we don't know in previous like our training. So it really can help us speed up this discovery process. For example, we don't know that this alpha beta mental core structure. Maybe for some domain expert they know it, but I'm guessing for all those people who are really new to these systems, it really can help discover some patterns because agent is really good at describing things that like hard for human to describe, especially for those cell interactions. Maybe we human cannot even verbally like make up a sentence to describe a lot of biology happening there. For example, I don't even know there's a word like core mental structure. Oh, and then the best part is that the agent can connect these word into some search queries and then pull online to see how people actually call it because agent also know a lot of like terminologies. For example, these maybe mesenchymal cell expansion, and maybe some non-expert don't even know. Okay, we can call this like phenomenon mesenchymal expansion. But I think the challenge basically here is that how actually to evaluate these results, you have to really collaborate with this domain expert. For example, maybe a lot of these things is really like convincing and all the literature's pool is like also evident, but we need to really final check with the domain expert to see if the agent is like doing really correct or is some like a point that he just hallucinate.
could I ask one follow-up question? Yeah. It's the output is English text, right? So it's interesting to think about like if there were some other output that you could do if it'd be able to, like, I understand that it's specific and now it gives you information that it can find, right? And that's how it develops that's how it knows something, right? Or it can explain something. It'd be interesting to see if there were other outputs that it could do. Anyways, sorry.
Currently implementing, like we are not only trying to give the text, we also want to render the images inside so they can be a more really like scientific paper like paragraph. So we can really try to help speed up scientific and scientists like workflow. Maybe just agent help you at least pre-screen a lot of like potential candidates. And actually, we are also trying to make more functionality to, for example, giving all this biological observation and let the agent to help you rank. For example, what's the most plausible hypothesis that you can take actions and what other people has been like. If this phenomenon is already well known, then maybe we don't need to spend too much time to study this. But if we discover something that is like less reported, maybe it's worth investigating.
Okay, terrific. Thank you, Mina. Guys, there are a number of questions I'd like to read to you that people have posed. I'll take it from the top. First is from Sharon Daniel, and of course you're welcome yourself to pose the question. Sharon asks about the flexibility of the tool. I mean, mainly what you showed was in Python, and the question is, can R tools be used in the space as well?
Yeah, I think we will definitely add more support for R, but in current version, we mainly use Python for several reasons. A lot of times, the LM itself is trained in a lot of Python code, so it's intrinsically better at writing Python code than in the R code. But definitely, R is also a very important tools for analyzing these omics, and we will try to make more support for R, like in the next work.
A potential solution for that is that there is a R2, RPY2 that you can connect R to the Python, which you write Python or inside Python. So potentially you can use that one to realize the same, but we are currently like exploring that. Yeah.
Or you can just have another agent convert it from Python to R.
Yes, there might be some potential overhead. So the best strategy maybe for Python integration is like more native because the agent itself is written in Python. But in R, we may need to scrape, like for example, parse the code snippet out and then put in R shells and then return a result back to agent. I think one challenge I can think is like because our agent really rely on the visual capability, so it means that it has to pass the image back to the agent's context memory. So Python itself like have a perfect I/O for image parsing, and R maybe I think we can do it, but probably with more engineering trick around.
So, a related question to that before I pose the next question in the chat is, I saw quickly when you were giving the demo a lot of text, which is great. Does it also give the pipeline outputs as well? Any markdown that?
Yeah, you can save the entire like conversation histories, or you can just prompt at the last, okay, help me summarize all my code. So it really depends on humans' need. Like
each party have the dedicated tool to show you what the code they are executing, so you will be able to access
it's
that.
very transparent and traceable. So if you find any potential mistake the agent may miss, like may make, and you can try to correct him. And because we save everything in the history, so it's really transparent.
So you're not having to rerun again. You can run chunks of the code just like you would if you had a large document and you're just going from one cell to another.
I think currently this part hasn't, because every time we like close the agent, all those variable will be
lost.
away. So, but I think it's doable. Like, but for that, I'm thinking because in the future it will be like as you see, like our current agent is more like a chat interface, but we are also working to like using the computer use agent like to really analyze the data. In that way, it's actually more native, like what a human will do, maybe open Jupyter Notebook and the agent directly code on Jupyter Notebook and then save everything. So,
terrific. There's a question in the chat about the depth of interaction with the literature. Are you searching full text? Is it abstracts alone? This is a question from Liz Butterworth.
Yeah, currently it's most about the abstract because you have this
publish
right issue, right? So, but another thing we are working on is to incorporate the parsing engine that can parse the PDF or other papers you download or you like parsed from the internet.
Yeah,
so
because internally we are using the Google Scholar Search API. So usually what we will find by using Google Scholar Search, that feature, the things you will find there, you can also find here. But a lot of things like definitely the some of the paper have those publisher rights, so it cannot have access to the full context. But I think that's probably not agent can solve because you really require the publisher to open access everything.
Could access like PubMed Central, which would be all of which would be the entire, and there are a lot of utilities.
Yes, that actually is we can definitely support that. It's actually also we are doing because like as you can see, like all these tools is really extensible. So
Sure.
it's also modular. You can just replace that part. The literature research tool itself can also be encapsulated into a smart deep research agent so that you can search at different different depths, and also you can do a lot of like literature re-ranking based
Yeah.
on the publication date. Maybe some of the more recent publication can have a more weight on the final results. That's like a lot of things that and we need to explore. And also we hope the community could also contribute and explore. Like we just like provide this concept, and a lot of these module can all be upgraded and improved, like the coding interface with the data and the literature web literature research with the different sources.
Yeah. Wenbo, Zuwan, there's another question from Ki Lu, who asks if this agent can run other spatial transcriptomics tools like cell communication. Essentially, how does it interact with other bioinformatics tools? Can you address that question?
Yeah. That actually is really extensible. So currently we equip them most with ScriptPy, ScanPy, and some tools that at least allows you to perform a lot of routine tasks. To allow for those cell-cell interaction tasks, we just need to kind of provide document, like coding document to the agent, just like what a human will do. If we want the human to learn to use a package, you just help provide a document for this agent, and then the agent can access the document and then learn to how to use these tools, and then they can provide the analysis on the data set. And we'll definitely during the revision we will address this.
Yeah. The beautiful part is the extensive framework, so you can leverage as many as you can, if not exceeding the capacity of agent to handle these tools. Yeah.
Yes.
That's great. Randy, do you want to ask your question? I think what you're doing is you're trying to confirm a particular understanding or a workflow. I'm happy to read it as well too. All right, let me read it to you guys. The question from Randy is, it's really cool concept. So you need to use Claude or OpenAI API to, okay, there you go. Randy, would you like to go ahead and ask? No, oh, you can't. All right, I'm sorry. So I'll continue reading. It says also, it seems like cell type annotation is done automatically by looking at your partial code. So structurally, you get the annotation markers from the literature. Then the agent runs Python by code generating the element, right? So I think it's just trying to confirm this this process. Can you comment on that?
I think we can have several ways of doing cell type annotation. Really depend on the complexity of your systems. And currently, we are working on those definitely agentic cell type annotation that the LM to do the cell type annotate, just like what we human will do. Like first do the clustering, annotate, and then give you the result. And also, we are also trying to integrate those foundation model type of annotation by transferring the label from the huge data set to the agentic system. And then ideally, we can combine these both source to provide more confidence of the cell type annotation. But we do think that like this type of annotation sometimes have some limitations, especially for very complicated system. Like we have a hybrid system, like a human islet implanted into the mouse kidney. So we directly have two species and two organisms. And sometimes even when the agent help you annotate, we still think it's better for human to do a final check and then consult the domain expert, especially in these systems, like it's probably wasn't trained in the LMs. It's like knowledge. Maybe sometimes it can hallucinate.
Yeah, and that's a question that I had for you as well, is about hallucinations and about controls essentially in your data. It was a beautiful. I enjoyed that figure that you showed where you were essentially putting an early and late image together, and then it was analyzing what it could see. Have you assessed by putting known understandings, I would say, of images and seeing if you could get rendered an appropriate understanding from what experts would say. Again, getting to this issue of controls and hallucinations. The other thing that struck me was how little input you provided to the agent at the initial prompt, and what amount of text and documentation that you got afterwards. And I'm wondering, is that because you've trained it very carefully to a particular few keywords? Do additional descriptions and prompts help it to become more careful? Because you didn't give an example of an hypothesis, for example, which would be quite interesting as well. So, few questions there.
Maybe I can first answer this hallucination. Yes, it definitely like all today's agent and LM, they are kind of prone to hallucination, and to really solve this or at least mitigate this problem is that's why we integrate a lot of these literature groundings. So that actually can at least confirm part of your reasonings like within the literature's grounding. So it's not crazy, and a lot of have some of the expert confirmations. And also another important point is that that's why we also need to really collaborate with the domain expert to help confirm maybe this phenomenon is like good, or this phenomenon is like plausible, or if you see something that's kind of really weird, so it's like probably not good. So we kind of at least have these guardrails to help us to trust the results. So I would think maybe in the future, what we human will probably do is to, based on these agent, propose some phenomenon or hypothesis, we can help evaluate if it is correct or not based on all our domain knowledge. I think at least at the current moment, we cannot hand off everything to the agent. And for the second part of the question, like why we use the small part of information and then the agent can do a lot of text, is that we generally give the agent kind of the scaffolding ideas like what we human will do to analyze data. So for example, for you to do first cell typings and plot the spatial plots, and also the the temporal dynamics, and also the neighborhood enrichment. So this kind of a general guideline could be provided to the agent as a general wanting to analyze, and the detail and analysis like execution, actually the agent can do it. And I think the most added value for the agent is actually coming from its visual inspection of those plots. So that part is really helpful because that's actually what we human also struggle the most of the time. Like actually, if we, I'm from a bioinformatics background, so the majority of the part of my work is not by actually writing the code, but actually writing the code is like not that difficult. It can do it, but the most of the part will like stare at those spatial plot and thinking, what does this mean? So this part is actually what the agent can really come in to help, and also linking this part of visualization back to its literature grounding and finding something like a hypothesis actually what the most added value of this agent.
Terrific. There's a couple more questions in the chat. Another question from Sharon asking about the local requirements of taking taking all the code that you've provided and then allowing one to spin it up locally. How many GPUs, that kind of thing. Can you address that?
I think so because the LM part is hosted on remote, because we are using the OpenAI, Anthropic, and other providers, we are not actually running the LM ourselves locally. So it's like you send your query and they send back a response. So that part we don't even need a GPU to do that. So, but regarding the size of data set, it depends on what data set you're working on. So it's just like you do the analysis on your own computer, what's the data set you can tolerate for running that. With the demo we showed that I just used my own MacBook laptop, which is run pretty smoothly. So I don't worry about the LLM part, but it's just about what data set you are working on for regular processing.
Yeah, so to give you a reference for that demo data set, we roughly have around 50K cells with around 200 genes. So basically, that size of the spatial omics can be very easily run on your local MacBook M2. So, but for the larger data set, we are also now exploring how to integrate with a cluster like compute. But the actually what agent mostly use is like the intelligence power because that part is hosted by the big company like Anthropic and OpenAI. So that will actually reduce most of the overhead. And so you just think this agent as like a borrowed brain from the big company to help you, like for them to sit in front of a computer to help you analyze your data.
Terrific. The last question that I'm going to read, I guess it's maybe not a question, but maybe an opportunity comes from Connor Finnerty, who points out, that conversation we were talking about. If you close the agent and you lose everything, are there?
Very, very good question. It's actually what we are doing. So like a long-term memory part of the agent is super important. And it's definitely we are doing like not only on semantic memory as Connor mentioned, but also want to also add the log memory, like what we human will like take notes. These are more like procedural or like episodic memory we human have. Like semantic memory probably is more like the biological knowledge we have. And those other memories like what we do every day, like the keep the log of that. So definitely its current version we haven't integrated that yet, but in the revision we'll definitely put more efforts in keeping this memory so it can be persistent across different sessions.
Connor, did you want to say something else?
Oh, I mean, I just I just want to throw out like I don't know exactly how you could integrate it into this specific agent, but I've just had one. I'm like kind of suspicious of what these companies do with your data if you don't run the LLMs locally, and then I also get sometimes frustrated with using generic LLMs, even trained LLMs that are available online. So I just like to do things that like retain information, across sessions. So I think if you were to integrate that into your tool, based off of my experience, it would probably work pretty well. That's my comment.
Yeah, that's a terrific suggestions. And for those privacy issues, we also have some. Yeah, but in our experience, like currently, like the most performance like model is still from those big company, and hopefully in the future, if we have more powerful open source models, we definitely be more than happy to use that because it can definitely provide more privacy, especially for those patient data. So like a pathology or patient like electronics like records, that part I think it's probably sensitive to those cannot be shared with the big companies. And we do see a lot of encouraging progress of the open source community like Queen or Llama. And these are now also be integrated into some chat or llamas, though it can be used, but in our current experience, it's still not performing that good, but I think this gap will be more and more closed in the future.
Terrific. Okay. Well, with that, gentlemen, I'd like to thank you for the presentation. Juan, Ju, I'd like to thank you also as well for this collaboration that you started. And we look forward to learning more about this, and the paper hopefully will eventually come out. Meanwhile, it is available through the bioarchives. We will have another webinar soon, so you can look for that announcement on the website. And with that, we thank you. And this recording will be available on YouTube, so you can check that out on our website.
Yeah, thank you so much, John.
Thank you guys very much. A wonderful time. Bye-bye.
Bye.